I appreciate the introduction. I will also hit on David's trial as I was a part of it, and hopefully it won't be too repetitive, that part. So hype, despair, and enlightenment, clinical trials journey, let's see, great. These are my disclosures, research support from a company called F2G. They work with antifungal drug development, not related, but they're a pharmaceutical company. Anson, which works on treatments for parainfluenza and influenza. And we tried out their drug early on in COVID during the desperate times, didn't work. So that's Anson. And then I'm advisor to Immunome, which is generally a company that works with oncology, but they tried and are working on developing passive immunotherapy to treatment of respiratory viral infections, including COVID. And then Carius is a diagnostic company. I'm going to start with a case because this is Hopkins, and everything is in service of patients at the bedside, although we can't take care of patients at the bedside unless we have a sense that we know what we're doing. So, and that's where you guys come in to help us identify therapies that can actually help people. So a 68-year-old man with history of lung transplant that I saw over the weekend. I was on call over this weekend. He has low antibody titers despite multiple COVID vaccinations. He was first tested positive for SARS-CoV end of December, just a few weeks ago. But despite treatment with remdesivir for 3 days around that time, his SARS-CoV PCR continues to be persistently positive. On January 24th, the cycle threshold was 23. That's a PCR cycle threshold. And when you're acutely infected, it's somewhere around 15 to 17. When you still have DNA in your nose, but or RNA in your nose, but you're no longer infectious or contagious, it's in the mid 30s. And so his was 23, suggesting that he still had a lot of virus in his body. And the question that was asked of us was, is there a role, when he was admitted to the hospital, for COVID-19 convalescent plasma in the care of this patient? And this question is quite distant from the study that we were involved in, but I think that the information that was gained from the studies that we did at Hopkins can be and is useful toward this this patient. And I'll go through our journey and then put together how we can use the information that we've learned here to help patients like this. One of the approaches in my mind to this pandemic is to really understand that it's evolving and dynamic, and that the conditions that we're seeing now are quite different than the conditions that we saw in March of 2020. And that doesn't mean that we need to start at square one. We can apply all the lessons that we learned to the patient that's really in front of us. So the basic principles of drug therapy, as you guys know well, are you got to give the right drug, you got to give it at the right dose to the right patient at the right time. And if you do those things, then there's a good chance that it'll work. Doesn't mean that it will work, but it really increases the chances that it'll work. So what am I holding in my hand? There I am in the blood bank, lovingly holding a bag of convalescent plasma. And it was a sort of an emotional moment for me to see this thing that we'd been working on as a real thing in the world. So you start with whole blood, and then you centrifugate it, and the plasma component comes to the top. The buffy coat is in the middle, and then the red cells, which is what makes the blood red, go to the bottom. So the top part is 55% of the total blood volume, and it's plasma, it contains a variety of different things, including beta carotene and bilirubin breakdown products, which is why it has the yellowish color. The concentration of yellow does not say anything about the quality of the antibodies that's in it, although I did have a patient and her husband get transfused as part of the study, and hers was yellower than his, and she said, I think I'm getting the real stuff. Not exactly. But the plasma component does contain antibodies, and if the antibodies are from people that have recovered from COVID, then there's a chance that those antibodies will then bind to the SARS-CoV-2 virus in the people that are infected, and therefore potentially prevent or treat the infection. So the rationale at the onset of trials is that passive immunization is an established approach to prevention and treatment of viral infections. So just a quick review. Active immunization is we give you a vaccine, some sort of product. That product activates your own immune system to then generate antibodies and T cells to protect you against the infection. Passive immunization is when you give preformed antibodies, and we do that all the time with rabies, people that have been vaccinated for rabies and then have rabies antibodies, hepatitis B immune globulin, RSV monoclonal antibody. That's a laboratory formed form of passive immunotherapy. Of course, we've seen a lot of monoclonal antibodies in COVID. Again, laboratory formed antibodies that do the same thing. They're passive immunization. And then convalescent plasma has been used successfully for hemorrhagic fever in Argentina, influenza, and success depended on a lot of factors in influenza, but overall was not as effective as usual therapy, but which includes antivirals, Ebola, and then bacterial and other viral diseases historically. So there was a historical presence for using passive immunotherapy for convalescent plasma. And here is a slide showing some of the background of that: Argentinian hemorrhagic fever, Spanish flu, SARS, the original SARS, old school SARS, and H1N1 flu in 2009 when it was called the swine flu, although that thought to have been originated in Mexico. Probably neither one of those was exactly accurate, but H1N1 used in all of those. Mixed results due to heterogeneous study designs, patient populations, and titers. So where in the spectrum of illness does passive immunotherapy for a respiratory viral infection, in particular in SARS-CoV, where does it work? So I'm going to take you through the journey of a patient. So you have the first part where a person encounters the viruses, and that's the post-exposure period. There's also pre-exposure, which is where vaccines really shine, but this is the post-exposure. Then there's the pre-symptomatic phase where they are infected but don't know that they're infected, and the problem with that is, as you can see on this slide, the amount of virus that's carried by the patient and then potentially transmitted to the person that's sitting next to them on the bus is quite high before they even know that they have COVID. Then once they know that, once they have symptoms, doesn't mean that they know they have COVID. They may do an antigen test at home and it's negative, and they have a false sense of security. This is what happened in our house, is that my son had symptoms of COVID, and he did an antigen test and was negative. So we all breathed a sigh of relief, saying, okay, well, we don't have COVID in the house. Who knows what other crud this is? And then a few days later, his symptoms got worse. He tested again. Now he was positive on the antigen, and now his dad, me, started developing symptoms. Well, I knew by then that I had COVID, so I tested myself, and lo and behold, my antigen was negative. Tested myself the next day, my antigen was still negative. Tested myself the third day, and my antigen was finally positive. You can imagine that if I had a job that required that I didn't have a lot of flexibility in terms of taking the time off, I might have gone to work infecting all the people around me. So so that has been a major form of transmission. But during this time, the disease is mostly driven by the virus. as antibody starts forming, the use of antibody-based therapy is probably not going to be all that helpful, because now the person is swimming in their own antibodies. And also at this time, the disease is driven by an over exuberant immune response. So the role of killing virus, and convalescent plasma is by and far an antiviral. It has some anti-inflammatory activity as well, but it's by and far an antiviral. It's probably going to become less and less helpful as the patient gets sicker toward hospitalization, shortness of breath, ARDS, ICU admission, and worse. That's by that time they have plenty of antibodies, so adding additional antibodies is not going to work. And the disease is driven not so much by the virus anymore, because as you can see, the viral genome level is plummeting, but by the overexuberant immune response. Which brings us to the cycle. So anybody have seen this Gartner cycle before? So I'll tell you a little bit about it. It was developed by a company named Gartner, and it has to do with technology and with use of technology by companies. And it can be applied to a lot of things. Right now, I think AI is a very hot area that it could be applied for, where initially there's the technology trigger, ChatGPT, and a lot of newspaper stories about it, a lot of excitement about it. And then people get very excited about it. And there's some early use reports that come in. Also, there's some situations where it didn't work, but gets a lot of hype, and people think it's going to make their teeth whiter and their clothes cleaner and solve world hunger. And you hit the peak of inflated expectations. Then you get the trough of disillusionment, where some of the trials come in and some of the information comes in, and it sounds like it's not going to be good for anything ever. We had that with convalescent plasma, in that there were early reports that it was effective in China, early reports from the US, where it was used as part of an emergency use in a sick patient, and they miraculously got better. Reports of Lazarus effects, where people were just dying, and they received the unit of or two of convalescent plasma, and they turned around and walked out of the hospital. Doctors at that time, there was nothing really to offer. You could do remdesivir if you were part of a clinical trial for remdesivir, or supportive care. And supportive care came in all different flavors, as people were quite stretched, so that you had ICUs not being run by ICUs, but by dermatologists, and so people way out of their level of expertise, and lots of mortality, both related to the uncontrolled infection and to the overstretched immune healthcare system response. So all over the country, people reached out to the FDA and said, we want to give convalescent plasma for our patients. And you can see all these different places where convalescent plasma was given, all the way from this little dot here in Arizona, which was in an area that had few health services to a lot of people, and a lot of people there had a lot of infections, to the East Coast. It was given everywhere. Some of the early information was that the product was pretty safe. So people were using it. Over 100,000 people received it. And one of the first lessons that came out of it was the stuff had to have high octane. If you gave stuff that had low titer, red, people died. If you had stuff that had higher titer, in blue, then people had a chance to survive. If you gave it here at 14.2% mortality versus 22.2, if you gave it once they were on mechanical ventilation, then then it was probably too late to give it, because we ultimately learned because the disease was driven not by the virus, but by the immune response. So important lessons here is giving it with high titer and giving it earlier on in the phase. A study from Argentina, randomized controlled trial, showed that if you give it to elderly people with high titer plasma within 72 hours of onset of symptoms, you can move the needle. If you gave them that same thing, but at a low titer, again, at if you were low octane plasma, didn't work. So give it early, give it with high titer. That was the early lessons that we learned. But then came the trough of disillusionment with this trial. It was part of the recovery platform, which was you guys have probably talked a lot about it in your center about these platform trials. They were very efficient at getting certain answers, and this was a open label trial of patients across the UK that were admitted to the hospital with COVID. And if you were in the hospital with COVID, you could be randomized to getting usual care or to getting high titer convalescent plasma. And when they did this in over 1,000 patients, didn't work, as you can see. There were certain subgroups that had some tantalizing suggestions, but given as a whole to hospitalized patients, didn't work. And that led to this big trough of disillusionment. And the combination of that study and a study out of India where they used low titer stuff, and a study out of Argentina where they gave it late and had low titer stuff, all three of those negative, and a certain political considerations because a candidate for president of the United States just before their national convention said that plasma is going to work and it's great, and then people reacted to the lack of specificity with that statement. All those things taken together led to a trough of disillusionment. The NIH didn't agree with the FDA. The FDA said yes, go ahead and use it. The NIH did not, and use of convalescent plasma plummeted significantly. Now, all this time, our group at Hopkins was working on trying to get us to this slope of enlightenment, learning how to use it, where it's going to work, where it's not going to work. And one of those studies was the study that I led, which is the post-exposure prophylaxis. So for both of our studies, post-exposure prophylaxis and early use at titer of greater than 320 using a certain assay. So we wanted to use higher titer convalescent plasma. We did not want to give stuff that had low octane in it. And donors had primary infection, and only high convalescent plasma use was permitted. This is the study organization. David, myself, and Kelly Gibo, executive committee. As you can see below, some of these patients are well known to you, including the DCC, which is your area of expertise that had made this trial possible. Site PIs across the US, and a lot of virology background, and a lot of blood bank background as well here in the plasma procurement, so that the stuff we gave was good stuff and safe stuff. Sites were selected because, as you may remember, during that time there were diverse hotspots. There were times when we had lots of COVID in Maryland, and other times when we had less COVID, but California was being hit with it, so we wanted to go all over the country, wanted to get diverse sites. They were diverse to a certain extent, but they were not diverse in that overwhelmingly these were academic, glass and steel, tertiary, quaternary medical centers. So that was a limitation of the work that we did. It was not very many standalone clinical trial sites that are embedded deep in the community. The challenges of the trial, in addition to the overall challenges using convalescent plasma and some of the lessons that I've already gone over, was also implementing a clinical trial during a pandemic. So very early on, just a lot of excitement. Yes, we can do this. This is going to be an easy trial. It's going to be like, hitting the side of a barn. And then the trough of disillusionment when we all find that doing a trial under pandemic circumstances after the initial excitement is really hard. And here's a quote from the remdesivir study, which we were not involved with, but I think it really tells the tale. So I won't read it to you, but you can kind of see some of the challenges of doing a clinical trial in that type of environment. And we had all those issues here. I remember one site that had a lot of excitement, which was in Florida. They had lots of patients, lots of excitement. They really wanted to be, they were our one non-academic center, and we were excited in having a non-academic center. And then they couldn't do it because they got swamped with COVID and the PI had to take care of patients. So these type of challenges. So here's the study: post-exposure prophylaxis. Subjects were going to be 500 subjects. Inclusions were adults that have had a high-risk exposure to COVID in the person in the 120 hours preceding their the time that they came into the trial. And it was somebody either at their home, at work, or potentially if they were part of a sports team, for example, and somebody on the sports team had COVID, or somebody at their office had COVID, or somebody in their home had COVID. They could all be enrolled as post-exposure prophylaxis. One-to-one randomization, 1 unit of plasma versus normal plasma. The reason that we used normal plasma as opposed to say normal saline was that plasma is a blood product and it requires certain procedures, and we felt that we would not be able to appropriately blind people if we had two nurses looking at which is what's required with a blood product, looking at the one product, and then sort of cursory looking at the normal saline, because they would know that it's not a blood product, and then that would tip things off. Also, it's hard to simulate the color of plasma. You can put vitamin multivitamin solution in saline, and that can look like plasma, but it's really not going to be the same. So we thought that we would not be able to blind it. And our lessons that we learned from the influenza studies that I was involved with about a decade ago were that if people knew what they were getting, it really significantly decreased the follow-up rate. If people knew that they had not gotten convalescent plasma, this was for influenza, then most of them didn't show up for follow-up visits, and we had problems with that. So there are problems with using a unit of convalescent of normal plasma as a control, because it's plasma, so maybe there's something in the plasma as opposed to in the specific antibodies that might make people better. So we've learned that no, if the plasma is not does not have antibodies, there's nothing magical about it. But as a whole, it seemed like it was the right thing to do, and after much discussion, we did that as the control. This is the study. So there was screening. Assessed for eligibility were over 2,000 patients. Excluded were 2,176 that didn't meet the inclusion criteria. At the telephone screening, did not meet the inclusion criteria after telephone screening, but when we talked to them in person, declined to participate. Randomized were 180. So as you can tell right off the bat, 180 does not equal 500. So we did not meet our enrollment target. And the reason for that, I think a variety of reasons. One is that people that were exposed to an infection were often unenthused in being involved in a clinical trial that involved a blood transfusion, particularly when they also had to take care of a family member who was now infected. So that was one issue. Another issue was that the trials were done in academic centers, and people were busy doing a lot of different things in academic centers, and didn't often have the bandwidth to really put 100% of their work into doing that. So at the end, we randomized 180. There was a third problem that came up that reduced our ability to randomize, and that's the development of vaccines. So vaccines were obviously a wonderful thing, and I hope that somebody gets a Nobel Prize or similar to that for development of COVID vaccines. For in my particular lens of the world, however, they created some problems in that one is if somebody had been vaccinated, it was hard to enroll them and expect that post-exposure prophylaxis with additional antibodies in addition to what they were going to develop from having the vaccine was going to help them in preventing illness. So somebody who's been vaccinated was that was an exclusion criteria. And also there was a statement put out by the ACIP, that's the recommendation group for vaccinations of the CDC, and that their recommendation was if somebody got convalescent plasma or monoclonal antibody, then they should not get vaccinated for the next 90 days. That impacted the ability of people that wanted to get vaccinated, which is oftentimes tracks with the kind of people that would want to be in a clinical trial. So many patients said came March of 2021, I don't want to get I don't want to be in this trial because that means that for the next 3 months I can't get vaccinated. And the CDC's rationale was that the antibodies in the convalescent plasma or the monoclonal antibodies might interfere with the vaccine uptake. So that was their rationale. I'm not sure that that rationale is scientifically accurate, but that was their rationale. And come March 1st of 2021, our enrollment declined to almost zero, and we had one patient in all of March 2021, and so we decided to shut things down, and ended up with 180 randomized as opposed to the 500 that we wanted. Half of those or so were allocated to control plasma, half to the convalescent plasma. Patients were followed up over the next 90 days, and ultimately ended up with 87 analyzed in the control group and 81 in the intervention group, the active product convalescent plasma with high titers. In terms of probability of infection, wasn't effective. We were not able to prevent the onset of infection, defined as symptoms plus a positive COVID test with either product. You can imagine the control product wouldn't do very much for it, but the high titer COVID convalescent plasma did not prevent infections. And if you only started counting after 3 days, you said, okay, somebody had exposure on day one to their spouse, sleeping in the same bed, spouse has COVID. Now the recipient, the study participant does not, but we enroll them, we randomize them, we get them going, and then day one after getting convalescent plasma, now they develop infection, we'd say, okay, well, maybe they didn't have enough time for the convalescent plasma to really kick in to provide protection. Let's say 3 days. So can we say only at 3 days does the clock start? Again, didn't make a difference. So bottom line was COVID convalescent plasma for post-exposure prophylaxis in this study was not effective. It was safe. The patients that got convalescent plasma, none of them had a severe transfusion reaction. There were actually more adverse events that happened in the control plasma, but they were mostly moderate. They were more adverse events, not statistically significant, but more adverse events in the grade 3 or 4 adverse events that happened in the control plasma. Some of that I can explain by things that had nothing to do with control plasma or convalescent plasma. Some patients got admitted to the hospital for treatment of chemotherapy for their bone marrow transplant. It had nothing to do with convalescent plasma, but there was an imbalance in safety. And hospitalizations for symptomatic COVID, there were none, and hospitalizations for other causes. Hospitalization for COVID-19, there were 2 in the control plasma, none in the convalescent plasma, but really no big difference in safety there. So that brings us to, again, the innovation, the trough of disillusionment. There we were, but we were starting to get a slope of enlightenment. We were starting to come up with a theory that for post-exposure prophylaxis, antibodies were not going to be effective. And that also tracked with some of the lessons that we learned from vaccination, in that that getting vaccinated wasn't necessarily going to stop somebody from getting COVID. It ultimately stopped people from getting severe COVID and hospitalization from COVID. But in terms of infection in and of itself, antibodies were not as effective as with some of the other viral infections, influenza and then measles, for example, where you can really prevent the development of infection at all. Polio, same thing with a vaccine. Not as much with COVID, and therefore giving antibodies to somebody who's had an exposure makes sense in that framework, that it wouldn't help. You've heard a lot about David's study, so I'm just going to race through it. This was an early treatment trial for treatment of patients that already had infection, and they had 1,000 patients, over 1,000 patients in it. About half got control plasma, half got convalescent plasma, and patients generally had outpatient type symptoms, all the symptoms I had with COVID, except my smell and taste. My smell remained, my taste not so much, and I didn't have skin changes, but pretty much all of these symptoms that people had as an outpatient for COVID. Half of the patients, again, got convalescent plasma, half got control plasma. And his study's outcome showed a significant difference between patients that got control plasma, had a higher chance of ending up in the hospital, than patients that got convalescent plasma. And in terms of outcomes, again, the main thing was hospitalization. That was really the main difference there. And the summary of the 2 studies was it did not prevent acquisition of infection when used as post-exposure prophylaxis. It did prevent progression of infection from mild to moderate to severe when used as early outpatient therapy, was well tolerated in both studies, and can be delivered in a safe and effective manner in an outpatient environment. In my study, we delivered it in the clinical trials unit that's located physically in the hospital at Hopkins and at centers around the country. In David Sullivan's study, it was done in a parking lot setting with tents and caravans. And so that brings us back to where in the spectrum of illness does convalescent plasma fit in. So I'm going to say that as pre-exposure prophylaxis, which is over here, we didn't test it. As post-exposure prophylaxis, we tested it. And I think even if we had enrolled 500 patients, if we had reached our enrollment goal, we wouldn't have really seen much of a difference because the efficacy, the difference in efficacy was so, there was no difference in efficacy. So I think even if we had ramped it up, we wouldn't have seen the split. Pre-symptomatic phase hasn't really been tested in those people that aren't yet symptomatic, but we know that they will become infected. So it hasn't been tested. Here, where in the outpatient setting, where there's high viral level, low antibody levels, this is where it's worked in a non-vaccinated, largely non-vaccinated population. That David Sullivan's trial and also the study by Libster from Argentina, and then what we've seen from the clinical trials in the UK and elsewhere is that once you get over here into hospitalization, shortness of breath, ARDS, ICU admission, doesn't tend to work. Now that's for a general population of patients. You can imagine that in 2023, where the population is largely vaccinated, and if they haven't been vaccinated, I think that there's vanishingly few people that have either never been vaccinated or never had the infection. So the interaction of the virus plus the immune system is quite different now than it was back in the day. So I would say that this line of antibody here is probably shifted to the left, because most people will have antibodies. So does passive immunotherapy in 2023 work for the general population? I would say probably wouldn't make much of a difference unless somebody doesn't have antibodies. And who's the person that's not going to have antibodies? That guy that I talked to you about in the very early slide that I saw over the weekend, the guy that has been vaccinated a zillion times and still has no antibodies because he's on mycophenolate and other immunosuppressive drugs for his lung transplant. Similar, somebody who might have gotten the drug rituximab, and somebody with other lymphocyte disorders. Those are the people that I think would still fit into this group of mild to moderate illness without antibodies. And those people also can be pushed a little further toward the hospitalization phase because they don't have antibodies or a good immune response. Their viral genome here, this curve gets pushed to the right in that they have high viral genome for a longer period of time. And in fact, here's a little bit of a touch of enlightenment from Senefeld, who pulled together all the articles, 3 randomized clinical trials, 5 matched cohort studies, 13 uncontrolled large case series, 125 case reports, put them all together and showed that COVID convalescent plasma in immune compromised people has a positive impact compared to control. I'm sure you guys can probably spend an entire semester teaching and arguing about the methodologies of this approach, but I think it is a little bit of a touch of enlightenment showing that for the immune compromised patient, an antibody-based therapy does still make sense. So here we are in winter of 2023, and I think we're in the slope of enlightenment, and that I would say that right drug would be an antiviral for somebody who could benefit from an antiviral, and that could be either Paxlovid or remdesivir or convalescent plasma, or if there's a new monoclonal antibody that became available that was effective against the circulating variant, that right dose, the if you're using convalescent plasma, it needs to have high antibody levels that work against the circulating variants. So getting material that has low antibody levels, not good. Getting material that was obtained during much earlier viral viral surges, particularly very early surges, is probably not going to be a good fit to the current viral viruses that are around. So that's the right dose. Right time needs to be done at a time when the disease is driven by the virus, not as much by the immune overreactivation response. Right patient, and I would say in winter of 2023, the right patient is going to be somebody that is immune compromised, who cannot receive other drugs. So the small molecule, particularly Paxlovid, I think that would be my first choice for most people. However, it has issues with drug-drug interactions because it has ritonavir in it, which interacts with many drugs. Remdesivir is another option, but it needs to be given at this point 3 days IV. Hopefully soon an oral version of that will be available. And then molnupiravir is the third small molecule option, and that's not very effective even in unvaccinated people, and in people that are vaccinated, it probably doesn't add very, very much. So in people that can't get Paxlovid or remdesivir and are in a place where they need antiviral therapy, because again, the vast majority of people right now that have been vaccinated 4 times, or how many times people have been vaccinated or have had infection, or a combination of both, they probably don't need anything. They'll get better. It'll be miserable, but they'll get better on their own. But the higher risk people that would benefit from therapy, and the higher risk people that can't get the small molecules and have an antibody deficiency, I think that is the slope of enlightenment. That's the right patient for treatment. Plateau of productivity, we've not reached it yet. I would say that there's many patients that fit that criteria that I just mentioned for convalescent plasma, but don't get it. Why don't they get it? Logistical reasons, ideological reasons. Some people are stuck in the trough of disillusionment and haven't quite learned from the slope of enlightenment, and they will tell patients or their staff, doesn't work, we're not going to give it. But again, I hope I made the case based on our studies and based on the totality of the data that there could be use scenarios, and that first patient, although that's not who we directly studied, the studies that we did have taught us about how it could be used in that patient. With that, I'll just finish with this acknowledgement, which is to the army of people that went on a limb and made these studies possible. And you can see them all here. And this huge group of people, including your members of your center who made this possible, and then the nurses who put their health on the line in seeing patients with risk for COVID and with acute COVID to make this study happen here and across the country. And with that, I'll take questions.
So Shmuel, first, it's Dave. I'm so sorry for having missed the very start of your talk. I'll help monitor some of the chat questions too, but I do have a question right now. What do you think that these trials have taught us about the use of convalescent plasma for the next pandemic? And how can we improve upon our ability to verify that they're safe and effective if we think that they are, and get them into use even faster?
Sure. So I think the main lessons that we learned is that convalescent plasma is first and foremost an antiviral. So as soon as we figure out with the next pandemic when the viral phases, as opposed to the other phases of illness, that's where you want to use it. Because it's an antiviral that works with antibodies, you have to figure out how to quickly qualify plasma so that the unit that's going into the patient is likely to have material that's going to be effective in them. And I don't think that we've actually perfected that even here with convalescent plasma for COVID, in that that patient that I was telling you about, which I gave them a couple units of plasma. I can't tell you for a fact that the units of convalescent plasma that I gave that patient had the right antibody to interact with the virus that they're actively infected, which is quite different than the way it works with small molecules, in that if I were to give this person some of the small molecule antivirals, I would have very high level of certainty that their virus would not be resistant to it. So we're still a little bit behind the 8 ball in that. I would say that the clinical trials, I look at clinical trials like a Formula One car. Unless they're very expensive, they're very flashy, they're and they need to run on an appropriate track, and unless the track is prepared carefully, then more likely than not they just end up in the shop. Sometimes they spectacularly crash, but oftentimes they just end up in the shop, and nothing exciting happened. So preparing for a clinical trial in terms of knowing what the patient population that you are studying, understanding their phenotypes, understanding the drug that you're giving, and then executing it, that's extremely important. In this pandemic, we didn't have that luxury, and oftentimes we like fools that rush in as opposed to the angels that fear to tread. We rushed in with randomized clinical trials, and the results were often difficult to interpret. And so I think really being thoughtful about how to do the clinical trial, understanding the patient population is super important. And I think that these studies give us information for the next one.
Okay, great. Thank you. Doug has a question. Go ahead, Doug.
Yeah, so I want to ask you a question about something I've speculated on without any real data. It's an upper respiratory infection, which means that IgA may be very important in terms of preventing infection. When you give plasma, you're probably giving IgG and IgA without secretory piece. And so possibly one of the reasons why it might not be effective in preventing the acquisition of infection is that you're not delivering the right antibody to the right site, whereas it's effective at preventing progression to severe diseases that, you're bathing the lung in IgG, which means that you prevent the severe respiratory manifestations. Just a thought, and any thoughts from you on that speculation?
You might be onto something. I will say that specific high concentrations of IgG have been effective in prevention for pre-exposure prophylaxis. For example, the drug Evusheld, which I can't pronounce the two components, was given effectively as pre-exposure prophylaxis when it was a good fit against the circulating variants. It's lost its authorization most recently because the circulating variants it doesn't work on. So, and although it's a monoclonal antibody, I think it's IgG-like. But I don't know, maybe the terms IgA, IgG, IgM, they fall apart when you're dealing with a monoclonal. So it could be that that in that way you're onto something. Those are my thoughts.
Okay, I think Sharisa Box has a question.
Good morning. How are you doing?
Hello.
Thanks for the great talk. You had mentioned something about some of the barriers for uptake. Could you expand on that? What's kind of stopping the immunocompromised community from accessing the convalescent plasma? And also, could you speak to the uptake globally as well?
Sure. So in terms of barriers, to give a unit or two of convalescent plasma, you need the following things to happen. So let's assume that I am immune compromised and I have COVID and I've tested positive. So now I need to have my blood typed. And so that's an interaction with the healthcare system. Now that I've had my blood typed, I need to, the blood bank needs to have a unit available. to fit with my blood type. Now it's just ABO typing. It's not the kind of complex blood typing that happens if you were to give me a unit of red cells or platelets, but it's still they need to have the right stuff in inventory. Then I need to come to a place, an outpatient place that will have 2 nurses staffing it so that they can make sure that they're not giving me the wrong unit. And then that's pretty much it. That's what it takes. Is it doable? Absolutely. Is it easier for my doctor to call in Paxlovid if I can get it? Absolutely. So those are big logistical hurdles. Now, if you take me to a country that doesn't have Paxlovid, that might be the only show in town. So it seems like it's worth doing. And in fact, in India, they did a great job giving a lot of convalescent plasma. In fact, they did such a great job that it ticked off the health authorities because they felt that it wasn't working. I think they were wrong in that actually outpatient treatment during the Delta wave in India was exactly the right use scenario. But so it's doable around the world. And it's been given in multiple countries around the world. Anywhere where you have a functioning blood banking system can do it. So one of the reasons why I got so excited about developing convalescent plasma, even when we were doing it for flu, was because if you have a factory that makes remdesivir, and that factory, most of the drugs made in this country are factories in Puerto Rico, which is sort of like a hotspot for hurricanes. So if you have a hurricane that knocks out the remdesivir factory, for example, you don't have any remdesivir. Or if you have a pandemic where all the workers at the remdesivir factory come down with COVID, you don't have remdesivir. So it seemed like this was sort of democratize it. And then also it doesn't have a patent. So you don't need these transnational agreements as to who's going to make remdesivir in India, who's going to make it in Ghana, et cetera. You can just do it locally. But the logistical hurdles are there.
Thank you. So would you say that the PR for convalescent plasma is pretty much recovered from all the negative press, or still kind of battling it?
I think that we are in the slope of enlightenment, but I don't think we've hit the plateau of productivity yet. So I think that there's the press is I view the press as tubes. What they're doing is they're amplifying what people are saying, and what people are saying, a lot of it was accurate. It's not going to make your teeth whiter and your clothes cleaner and take care of everything. It needs to be used correctly. And what is correct use in April of 2020 is not correct use in April of 2023. So it needs to make an adjustment, which is why the patient I presented was nothing like the kind of patient we were thinking about back in April of 2020.
Thank you.
Yeah, thank you so much. So we have run out of time here, although Shmuel, we did have one more person who had some questions. If you have a moment to stay on.
Sure.
Thank everyone for joining us today, and especially thank you to Shmuel for a great presentation.
Thanks. What, you think you're going to shut me up about convalescent plasma? I'll be available as long as needed.
Oh, well, maybe you have a patient to infuse. So we had a student who, I think a student, I'm sorry.